Alright, so this lesson is going to be on DNA replication, and it's running parallel with the book. Uh, it's section 12.3 in your textbook. So, you're going to be doing reading notes on 12.3, but this is just to get a little bit ahead since you guys will be going on spring break. I wanted to introduce this before you guys left. So, the daily objectives. So, for this lesson, you should be able to, at the end, summarize the events of DNA replication and compare DNA replication in prokaryotes with DNA replication in eukaryotes. So, copying the code. So, base pairing explained how DNA could be copied. Base pairing again was the A with the T. Remember, adenine always bonds with thymine. Cytosine always bonds with guanine. That's Chargraph's rule. Okay. Um, so base pairing explained how DNA could be copied or replicated because each base on one strand pairs with only one base on the opposite strand. And each strand of the double helix has all the information needed to reconstruct the other half by the mechanism of base pairing. So basically, if we know what one side is, we know what the other side is going to be. So we can take either side and make a new DNA strand, if that makes sense. If we have one side that's ATA in that order, then the other side has to be TAT, right? So if we have either of the sides, we can come up with what the other side is because there's only one other possible combination for each of the bases. And that's going to come into play when we talk about replication. So since each strand can be used to make the other strand, the strands are said to be complementary. And I'm going to use that word a lot. So complementary strands. Okay? And it just means that they are essentially opposite. They're opposite, but they can be used to make the other strands. So, before a cell divides, it duplicates its DNA in a process called replication. And we know that this happens during the S phase of interphase, right? So this process ensures that each resulting cell has the same complete set of DNA molecules. And we're going to go into how exactly it does it. But this image is actually really, really good. I also have a video that you guys are going to watch that I think explains it really well. So during DNA replication, the DNA molecule separates into two strands. Okay, so you can see, you can see how where it says replication fork, that's where the DNA is splitting. Okay, so each side is splitting off, and then where they're splitting off, there is a new strand that's being copied on either one. Um, so it separates into two strands and then produces two new complementary strands following the rules of base pairing. So A with T, C with G. Each strand of the double helix of DNA serves as a template or model for the new strand. So, as the result of one strand splitting off into two, we're gonna eventually, at the end of this, make two identical DNA strands, right? So the two strands of the double helix separate or unzip If you think of it like a zipper, it actually makes sense really, really well, because the if you think of the DNA together as one thing, is the zipper completely zipped up, and then as you unzip it, you get two complementary strands. So as it unzips, it allows two replication forks to form. So this would be kind of like if you broke your zipper in the middle of it, and it was zipped up on the bottom half and the top half. I hope that makes sense. I hope I didn't confuse you there, but um, very similar to the image that's shown. So, as each new strand forms, new bases are added following the rules of base pairing. If the base on the old strand is adenine, then thymine is added to the new forming strand, and guanine is going to bond with cytosine. Okay? Now, the result of replication is, like I said, two DNA molecules identical to each other and to the original molecule. As you can see, you know, once we make that fork, we start to make that other strand, and we're starting to get two identical DNA strands. Um, each DNA molecule resulting from replication has one original strand and one new strand. So we're finishing with two, which is going to be important if we are doing cell division. So the role of enzymes. So we can't do this without enzymes. And you might be saying, like, how are we just making a new complementary strand on each of the unzipped strands? So this is done by enzymes. So DNA replication is carried out by a whole bunch of enzymes. We're really only going to focus on one, but there's actually several. But first, there's an enzyme that unzips a molecule of DNA by breaking the hydrogen bonds between the base pairs and unwinding the two strands of the molecule. So then each strand then serves as a template for the attachment of complementary bases. So that's nothing new. Now, the principal enzyme involved in DNA replication, and really the only one that we're going to talk about, is called DNA polymerase. So you need to know that, okay? And you need to know what it does. So DNA polymerase joins individual nucleotides. Remember, a nucleotide is just one little ladder of DNA. So it's the phosphate backbone, the deoxyribose sugar, and the nitrogenous base. Um, so it joins individual nucleotides to produce a new strand of DNA. So DNA polymerase also proofreads each DNA strand, ensuring that each molecule is a perfect copy of the other. So really, really important there. It is actually what's it's reading the DNA, and as it comes across an A on the unzipped strand, it puts a T there. And then it might run into a C, so it puts a G. Then it runs into another A, so it puts a T. Then it runs into a T and it puts an A. So it's making the complementary strands as it goes along, and then if it makes a mistake, now a mistake might look like putting a C with an A. Now we know that only a T can go with an A, right? A always binds with T, C always binds with G. So if A was accidentally bonded with a C, then DNA polymerase would see that mistake, take it off, and fix it and put the T there so that the A binds with T. That's what it means by proofreading. So the next thing that I want to talk about is telomeres. So the tips of chromosomes, and remember chromosomes are just coiled up, densely packed DNA. Um, So the tips of the chromosomes are known as telomeres. So the ends of DNA molecules located at the telomeres are particularly difficult to copy. And over time, DNA may actually be lost from telomeres each time a chromosome is replicated. And telomeres actually have a lot to do with cancer. We're actually looking into the length of the telomeres in association with cancer cells. And there seems to be a correlation there. But it also shows that older cells will actually have shorter telomeres. So what they do to cells actually have a method of combating this. So an enzyme called telomerase compensates for this problem by adding short repeated DNA sequences to telomeres. Now, these DNA sequences don't actually code for anything. They just are there so that if something gets cut off, it's not important. So it lengthens the chromosome slightly and it makes it less likely that important gene sequences um, won't be lost from the telomeres being cut off. So an example of a telomere might be like T, like at the very end of the DNA strand, it's probably going to say like T, right? And that doesn't code for anything. It's just a bunch of thymines next to each other. Um, and then the copy would, or the complementary strand obviously would say A, right? So that doesn't code for anything, but if a couple of those T's and A's get cut off, we're not losing any important genes. So that's why that's there. So, replication in living cells. So, the cells of most prokaryotes, now we're talking about prokaryotes, so like bacteria, remember, don't have a nucleus, have a single circular DNA molecule in the cytoplasm. So, circular DNA, super important, containing nearly all the cell's genetic information. Eukaryotic cells, on the other hand, can have up to 1,000 times more DNA. Nearly all of the DNA of eukaryotic cells is found in the nucleus. So, a little side note, it becomes super, super easy to map like bacterial DNA and also to do experiments on bacterial DNA as opposed to human DNA because human DNA is just so, so much bigger. Um, Okay, so in most prokaryotes, DNA replication does not start until regulatory proteins bind to a single starting point on the chromosome. Okay, so you have these regulatory proteins, um, and what they do is they have to actually bind to a part of the chromosome in order to start DNA replication. Okay, this actually will trigger the beginning of DNA replication, and it's a little bit different. So we're gonna take a look at it because it's in that circle, right? We have that circular DNA, and there's a good image that kind of shows it. But replication in most prokaryotic cells starts from a single point and proceeds in two directions until the entire chromosome is copied, right? Until we have two identical circles of DNA. So this is an example of prokaryotic DNA. So often the two chromosomes produced by replication are attached to different points inside the cell membrane and are separated when the cell splits to form two new cells. Not much there. You can see the difference here is that we have really just the circular DNA and that it's just making the complementary strand right next to it, and then once the cell splits, that's when the DNA actually splits. So eukaryotic chromosomes, on the other hand, are generally much bigger than those of prokaryotes. And we know that because eukaryotic cells are much bigger than prokaryotes. But in eukaryotic cells, replication may begin at dozens or even hundreds of places on the DNA molecule, and it does proceed in both directions until each chromosome is completely copied. So you can see the example here that we have multiple replication forks, and the DNA being replicated not just from a single fork happening, but actually all along the DNA strand until the entire thing is copied. So that's actually different from prokaryotes, where it's only going to happen at one spot. So the two copies of DNA produced by replication in each chromosome remain closely associated until the cell enters prophase of mitosis. So, what happens in prophase of mitosis? Well, we know that the chromosomes condense and the two chromatids in each chromosome become clearly visible, right? That's when we can see it as like an X, right? The chromosomes, we can see them as an X because they have the two sister chromatids joined at the centromeres. Uh, they separate from each other, right, in anaphase of mitosis, and then it produces two new cells, each with a complete set of genes that are identical and coded in the DNA. So, this first video I have um, on the Shobi assignment, and that's what I want you guys to watch next, okay? The second one's really long, so I didn't post it on there, but if you guys are interested, you can find it. It's Bozeman Science, DNA Replication. Just search it up on YouTube and you can find it, but I think it's like 12 minutes, so I didn't assign it. But it's pretty good. So the next thing that I want you to do after you do the guided notes to this and you're all done, watch that video. It's short, it's maybe 3 minutes, and it's really, really good because it just sums up in a really good animation how DNA replication actually occurs, like step by step. And that's personally like how I learned it. It's just from watching animation um, of DNA unzipping and the DNA polymerase coming in and synthesizing a complementary strand and proofreading. So it's a really, really good resource that I want you guys to check out.